The most precious thing is that you still love science.
And know that you belong to me.
We're all in this journey together.
Have fun with what you do.
No matter what you do, make sure that you share your science with everyone. Make it accessible.
Always ask questions no matter what.
Know that you can learn something from anybody.
If you want to go fast, go alone.
Dream big.
If you want to go far, go together.
Be curious.
Hello, and welcome to the 2021 SCIEX Innovation Showcase. Last year, we celebrated our 50th anniversary with landmark products, capturing 50 years of innovation and experience in the quantification of molecules. Those landmark products are now enabling customers to break throughput bottlenecks and, in many cases, exceed current limits of detection for LC-MS. Our gaze is now firmly fixed on the future, a rapidly changing future where the expectation of science is ever increasing. It's clear that things need to be done differently, that incremental improvement is not enough. Only step change and innovation will meet the increasing demands of the future. Today, we are here to introduce the Xenotolf 7600 system, a new platform and mindset in accurate mass LC-MS that has the power to change people's perspective.
A revolution in accurate mass has arrived, where new thinking and new possibilities provide new routes to extraordinary answers. Introducing the SCIEX Keynote 7600 system that's rewriting the rules. Now, looking from a new perspective puts what you knew into perspective. Welcome to the Xeno revolution. Discover new levels of control with a step change in fragmentation technology. Through high-energy collisions, you can create reproducible fragmentation patterns. Or precisely disassemble molecules, exploring new perspectives. Fine-tune fragmentation energies, allowing controlled electron-activated dissociation of all molecule types. Explore advanced fragmentation mechanisms that create a myriad of insight within your reach. Xenotrap pulsing unlocks extraordinary sensitivity gains, uncovering information that has always existed undetected, adding a brand new perspective to your quantitative and qualitative data. Watch as rare data becomes your everyday. Now your toughest challenges become your greatest advantage. Now new discovery is not only possible, but quantifiable. Now you have the power to choose how. Now you can be extraordinary. From the leaders in quantification of molecules.
We believe this system delivers that crucial step change in MS-MS technology, unlocking new, precise perspectives on any given molecule that will impact lives in a profound way. Powering understanding to better and earlier diagnosed diseases, powering access to personalized therapies and biotherapeutics. A powerful leap in proprietary innovation, xenotrap pulsing, and electron-activated dissociation will reveal new, rare, or even previously undetected information on an everyday basis. We are also proud to announce key advances in software that will make the Xenotolf 7600 more accessible globally, connecting every customer with new technology and with one another. As an innovator, we need to fully understand the problems that, if solved, would enable future breakthroughs for our customers. Yet this future and all of its breakthroughs do not exist in the mind of one person. It is a future powered by diversity of thought, approach, and identity. It requires a multitude of different viewpoints and a cultural shift that ensures those viewpoints are heard and included. When empowered in this way, diversity allows innovation in ways we could have never thought of before, enabling a new generation of scientists to meet their potential. It is their talent that, when harnessed, is what will define our future. It is the power of difference that drives us all to be extraordinary. And so, the Xeno revolution has begun.
We're extremely excited about the potential of this new accurate mass product and the impact it will enable our customers to make. But to be here today, launching this product has been a journey. To gain insight, we spoke to hundreds of customers globally. We also engaged our Innovation Advisory Board, made up of thousands more. Together, we explored the real-world limitations of current MSMS sensitivity and ion fragmentation techniques. They told us these limitations were holding back the process of bringing critical drugs to market, holding back the development of advanced next-generation therapies, and holding back the discovery of new markers for cancer and other life-threatening diseases. It is through listening to these customers and harnessing their perspectives as a blueprint for innovation that the Xenotop 7600 system was born. New perspectives and information uncovered by the Xenotop 7600 system will be the basis for our customers and a new generation of scientists to execute their extraordinary visions. Our customers working in cancer research told us that they couldn't characterize the post-translational modifications of low-abundant proteins such as transcription factors and their binding partners. They told us this was preventing them from finding novel drug targets for cancer therapies. We thought differently. We engineered a system with EAD fragmentation that can routinely characterize PTMs with the sensitivity to study these critical low-abundant proteins. Our customers tell us that for the first time, cancer researchers will be able to develop new therapies that target these proteins and understand the role they play in oncogenesis. Customers studying inflammatory diseases told us that they were unable to discover specific lipids markers due to poor MSMS specificity. They also told us that the sensitivity challenges posed by signaling molecules meant that they couldn't precisely measure disease-specific differences in cancer and autoimmune disease. We thought differently. We engineered a system that can differentiate between conformational lipid isomers at the MSMS level with EAD, a system that could detect, characterize, and quantify species at low levels with the sensitivity of xenotrap pulsing. Our customers tell us that for the first time, individual lipids, rather than the entire class distributions, can be studied as potential markers for a whole host of diseases. Biopharma customers developing next-generation therapies told us that the existing approaches in detection and prevention of potentially fatal changes during production were taking too long. This then delays bringing these life-changing drugs to market and potentially impacting their safety. We thought differently. We engineered a system that can differentiate between leucine and isoleucine at the MSMS level in a single run with XenoEAD, and the capability to precisely pinpoint the site and composition of glycan modifications. Now, novel complex therapies can be developed faster and with added confidence in their safety. Innovation is at its most powerful when it creates opportunity. The opportunity to see new perspectives that drives life-changing discoveries.
The Xeno revolution is brought about by three key innovations: xenotrap pulsing, electron-activated dissociation, and SCIEX OS software. The impact of these new technologies is the ability to extract more information from every sample. In the past, duty cycle limitations of time-of-flight technology meant that 90% of ions were thrown away during the process of analysis. Our customer told us that this limitation meant that low-abundant, biological significant species couldn't be detected, preventing them from making breakthroughs in disease research, biomarker discovery, and new novel therapies. With xenotrap pulsing, we thought differently. Ions are accumulated in the xenotrap before being pulsed rapidly into the TOF, meaning we can detect up to 10 times more ions. Consequently, each TOF experiment contains more useful MSMS information, particularly on low-abundant species that were previously undetectable, introducing our customer to a new sensitivity revolution. They will begin uncovering structural information previously inaccessible and drive the limit of quantification achievable with accurate mass. They will also be able to detect new multi-omics markers for disease for the first time and characterize the metabolic pathways of new drug therapy in order to assist with patient safety. Xenotrap pulsing gives the ability to detect lower abundant ions. Many customers were using alternative fragmentation techniques such as ECD and ETD when trying to uncover crucial structural information. Yet these techniques were only useful infrequently and in specialist situations. They had no ways to controlling fragmentation energy, limiting their capability to study peptides, lipids, and large intact proteins. The electron energies of these techniques were insufficiently to break high energy bonds in the way required to study small molecules for drug discovery or metabolomics. With EAD on the Xenotop 7600, we thought differently. EAD technology allows the user to fine-tune the electron energy, accessing multiple fragmentation mechanisms that can be applied to large molecules, peptides, lipids, and small molecules. EAD technology is not a fragmentation technique. It is a spectrum of tunable fragmentation techniques delivered in a single platform. Now our customer will be able to choose fragmentation energies that are specific to their molecules of interest, enabling them to acquire key MS/MS features that are needed to identify post-translational modifications, lipid structures, or critical quality attribute in the development of biologic drugs. Combination of these innovative technologies will provide information that opens new doors to new ways of working and give new insights into biology, medicine, and safety, all enabled by the Xeno revolution.
Software is a vital connector between technology and insights that will drive discovery. Today, we are proud to announce 3 new products and software that will make these discoveries accessible to our customers. Firstly, today's launch of the Xenotop 7600 means that all of our flagship LC-MS platforms are now powered by SCIEX OS. SCIEX OS will be the jump-off point for an entire software ecosystem with constantly improving data processing algorithms. It will be the future hub for all analyses to ensure completeness and consistency across all your labs, instruments, and workflows. Within the next 12 months, we will be launching SCIEX OS in 9 different languages to create a truly global software ecosystem, allowing many people, regardless of location, background, or experience, to connect with the new technology and produce meaningful impact. Today, One Omics Suite becomes a commercialized product that will enable big data approaches through the power of cloud computing. Our customers told us that the new horizon in disease research is multi-omics, the ability to connect genomics, proteomics, and metabolomics data together and create new, complex insights within biology and precision medicine. One Omics Suite will enable life science researchers to process large-scale multi-omics datasets, making breakthrough discoveries in the field of biomarker research, aided by the ability to globally collaborate with other researchers. Our new Molecule Profiler app, launching today, will enable a comprehensive understanding of low-level in vivo metabolism. The new frontier of therapeutic modalities is oligonucleotide-based therapies. Therapy based on DNA and RNA that can act directly upon the process of gene expression. Our customers told us that the complexity of these molecules during production and the way they metabolize presented a critical challenge in making these drugs safe for use. The Xenotop 7600 is uniquely capable of detecting low levels of oligonucleotide metabolites in biofluids using sensitive negative ion xeno-CID approaches. Combining this with the ability of the Molecule Profiler app, it becomes easy to correctly identify low-level metabolites and understand their in vivo metabolism, ensuring that these extremely effective therapies can be brought to market faster. In the future, a catalog of apps, like Molecule Profiler, will allow users to build their own software processing pipelines within the SCIEX OS ecosystem, according to their individual workflow needs. For the first time at SCIEX, we are creating collaborative, responsive software that prioritizes user experience and diversity of need, to enable the vision of our customers.
It is our customer insight and extraordinary vision that drives us. But how do we exceed their expectations and become a force for good? We're challenging ourselves to take a step further when looking to the future, considering the need to nurture a fresh new generation of breakthrough thinkers who have the potential to take these innovations to new levels. At SCIEX, we believe we have a responsibility to inspire and enable the next generation of analytical scientists, acknowledging the challenges that they all face, as well as the unique challenges experienced by some because of their identity, gender, or ethnicity. Diversity needs to be at the heart of this generation. But to achieve great things, diversity needs to be more than just a word to us. It must be the blood that runs through our veins. Listening is key to understanding how and where we can create change. So this year, we asked the global scientific community for their advice. We asked them to become mentors and role models to a girl whose story we created, an extraordinary girl called Grace. For us, Grace represents the future. She represents the talent and enthusiasm that is present in thousands of young people who just need guidance from the generations before them to fulfill their potential. This initiative will help us to understand the challenges faced by our underrepresented groups building a career in science, and the role that we can play in creating opportunities and role models for them. We want to work for the next generation as well as the current, to work alongside them and have them work for us. We want to enable them to share their vision for the future alongside everyone else's and use this vision as a building block for innovation. We want them to harness products like the Xenotov 7600 system to empower their vision, their potential, and their perspective, to affect the lives of the current generation, the next generation, and the generation after that. Only through listening and learning and experimenting can we really understand how to move forwards. Our vision and our customers' vision must continue to be inextricably linked. And so, the insights we gather from all customers become the building blocks for what we can achieve together. They are what sets our bar and what drives our desire to exceed it. It is the power of difference that drives us all to be extraordinary.